So, hello dear students, welcome to the next session of Principles of Inheritance and Variation. Today we are going to discuss regarding some other examples for exceptions to the Mendelian principles. So, next such phenomena is Pleiotrophy. So, what do you mean by Pleiotrophy? As we know that from the Mendelian principles of inheritance, one gene code for one character, or one gene code for one trait of a character. But here, a single gene product that produces more than one effect can also be called as the single gene product which controls several phenotypes depending on its position. So such a phenomena where the single gene or single gene product produces more than one phenotypic effect, such phenomena is called as pleiotrophy. So what do you mean by pleiotrophy? The single gene product producing more than one phenotypic effect or producing more than one effect, that is called as pleiotrophy. Then what is the basis of pleiotrophy? Why single gene produces more than one effect? So the basis is nothing but the basis of the pleiotrophy lies in interrelationship between the metabolic pathways that produces different type of phenotypic effects. Means various metabolic reactions which takes place in our body, the product of one metabolic pathway may be the substrate for the other metabolic pathway. If the one metabolic pathway is not functioning properly, means then the particular product will not be formed, so that the next pathway will get disturbed. As a result, some other product or a defected product may be formed. That's how different type of phenotype will be produced. So, interrelationship between the metabolic pathway is the main basis of effect called as the pleiotrophy. Let us understand this pleiotrophy with examples. See, this definition is very important for the PU examination, and examples as well as their explanation is important for the NEET examination. Let's see. The one of the example for the pleiotrophy is phenylketonuria. So what is phenylketonuria? It is an autosomal disorder. Later you will get to know what are autosomal disorders. Okay. So which disorders are sex-linked one? What do you mean by autosomes? What do you mean by sex chromosomes or allosomes? So, what are the disorders which are transmitted from parents to the offspring through chromosomes? That we will discuss at the end of this chapter. Okay. Now, just remember, phenylketonuria is an autosomal disorder. See, mutation in a gene which code for the synthesis of an enzyme called as phenylalanine hydroxylase. A particular gene which code for the synthesis of phenylalanine hydroxylase enzyme. If that gene undergoes mutation, means when the gene is in normal condition, it code for the synthesis of phenylalanine hydroxylase. So what is the function of this enzyme? This phenylalanine hydroxylase which converts the phenylalanine, an amino acid, into tyrosine. Its function is to convert phenylalanine in our body into tyrosine. That is what its normal function is. Then that individual will be normal, having normal phenotype. That is one effect of that gene. Means when gene code for the synthesis of phenylalanine hydroxylase, the person will be normal. That is one phenotype. But if the same gene undergoes mutation, means changes in its sequence or the genetic information, if it gets changed, means that is called as mutation. So, the mutated gene then fails to synthesize the phenylalanine hydroxylase enzyme. So, when this enzyme is not produced by mutated gene, then what happens? Phenylalanine, instead of tyrosine, it gets converted into phenylpyruvic acid. Phenylpyruvic acid. This phenylpyruvic acid, when it gets accumulated in our body, that leads to the phenotypic effects which are characterized by mental retardation. There is a loss or the reduction in the hairs, and also the reduction in the skin pigmentation or skin coloration. Skin color reduce ಆಗುತ್ತೆ, hair loss ಆಗುತ್ತೆ, that person will become mentally retarded. Okay, and he will pass a black colored urine. So these changes in the phenotype of that individual is because of a mutation in the gene. Means when it is present in normal condition, the gene produces normal phenotype of that individual. When it get mutated, it produces other phenotype. So same gene is controlling or producing more than one effect. Such effect is only called as pleiotrophy. Here gene is one only, but based on its position, it produces different type of phenotypes. So one gene controlling more than one effect or more than one phenotypic effect is called as pleiotrophy. So that's how the phenylketonuria is an example of the pleiotrophy. Make a note of these points now. Later we'll discuss some other examples of the pleiotrophy. So the other examples of the pleiotrophy are as follows. So one you can observe in Drosophila. What do you mean by Drosophila? Means a small fly, or the tiny fly, which sits over the fruits, and you will be very often you can see these Drosophila in kitchen also, right? Over the organic or the leftover in the kitchen, or the leftover food in the kitchen. There you can see a small sized flies. Those are only Drosophila. So the common name of the Drosophila melanogaster is fruit fly. See, the mutation in the gene which is coding for the white eye in Drosophila, that also leads to the depigmentation in other parts of the body. The gene which code for the white eye, when the gene is normal and coding for the white eye, remaining parts are also will be in normal color only. But when that gene coding for the white eye get mutated, that leads to other phenotypic effect also, that is a depigmentation in other part of the body. So here the single gene coding for two different phenotypes. So it is also example of pleiotrophy. Similarly, let's understand one more example. Say for example, a gene which code for the control of the starch synthesis in the garden pea plant. Gene which is coding for or which is controlling the starch synthesis in pea plant. One gene means this gene is having two different alleles, that is one is dominant allele and another one is the recessive allele, that is capital B and then the small b. Let's see what happens here. In homozygous condition, that is capital B and capital B, when this allele is present in homozygous condition, synthesis of the starch will be efficient, so that the seed size will be a large or the longer. Larger or longer. But when another allele which is present in homozygous condition, starch synthesis is very less and seeds are found to be very small sized or the short sized. So after maturation, the seeds with the allelic combination capital B and capital B, that is homozygous combination of capital B alleles, they were found to be round. The seeds were found to be round. But the another homozygous condition, that is of small b, when these alleles are present in homozygous condition, seeds were wrinkled. Seeds were wrinkled. So here homozygous condition or homozygous condition of the capital B allele, that leads to large sized seeds. Upon maturation they will become round seeds. But the homozygous condition of recessive allele, that is small b, here what happens, the seeds will become wrinkled after maturation. But when the other condition is considered, that is heterozygous condition, capital B with the small b, then seeds are found to be round only. but their size was very intermediate. Means what is expected? If capital B is there, if it is considered to be dominant allele, and when it produces a round shape, when present in homozygous condition, if it is dominant means it must be producing a large sized seed itself. But only for the shape it shows dominant, but for the size it is incompletely dominant. Let us understand it with the representation of that grass. When the plants, pea plant having the alleles which code for, so here what happens, the round and large seeded plant were crossed with, here what happens, wrinkled and small sized, wrinkled and small sized seeded plant. So this generation is considered as a parental generation, and when they produce the gamete, so only one type of gamete will be produced as they are homozygous. So this generation, these are gametes. So when the fertilization takes place between such two gametes, then they produce us the offspring which is called as F1 generation. And all the F1 offsprings were found to be with, so these are heterozygous. So that what is the phenotype of the heterozygous here? Shape will be round but intermediate sized, right? The round and intermediate sized seeds, intermediate sized seeds were produced in F1 generation. When such F1 plants, they are allowed for selfing or self-pollination. So what happens? Then they produced the offsprings and their ratios or phenotype was found to be. So ಏನಿದೆ ಹಾಗಾದ್ರೆ? So here the gametes produced, one is dominant allele containing gamete, other one is the recessive allele containing a gamete. Similarly, the one more parent also produces dominant allele containing gamete, recessive allele containing gamete. At this stage what happens now? Let's write the F2 progeny now. F2 progeny or F2 offsprings. So what is the combination here? Capital with capital means homozygous condition. Next, this dominant allele with the recessive one, so heterozygous. Dominant one with recessive, heterozygous condition. Recessive with recessive, homozygous recessive condition. Yes, let's write the phenotypic ratio and genotypic ratio now. What is the phenotypic ratio now? Phenotypic ratio. Let's write phenotypic ratio, okay, and genotypic ratio for the phenotypes separately. So here for seed shape, what is the phenotypic ratio here? When a single dominant allele B is present, whether in heterozygous condition or the homozygous condition, if a capital B is present means those seeds will be round shaped. So how many are round here? 1, 2, 3. So 3 are round and the one is wrinkled. 3 are round, 1 is wrinkled. So what this 3 refers means here, 3 is wrinkled and 1 is 3 are round and 1 is wrinkled. So 3:1 is the phenotypic ratio. Then what about genotypic ratio here? 1 is heterozygous dominant for the roundness of seed. 1 is heterozygous dominant. 1 is homozygous dominant. 2 are heterozygous. And one more is homozygous recessive. So, genotypic ratio is 1:2:1. That is 1:2:1. Right? So, phenotypic ratio is 3:1. Genotypic ratio is 1:2:1. It is same as that of Mendelian principles. Where there is an exception in this, we have considered this one as an example of exception to the Mendelian principles, right? Is there any deviation from the Mendelian principles? In Mendelian experiments were also phenotypic ratio was 3:1 and genotypic ratio was 1:2:1. It is also same here, right? But if the other phenotype is considered at a time, then what happens? When the other phenotype, that is for seed size, what is the phenotypic ratio? Seed size, the phenotypic ratio, both homozygous dominant are present means what happens here? The size will be large, and then when heterozygous condition is present, the size of the seed will be intermediate. And when homozygous recessive condition is observed, then the size will be small. Seed size will be small. So in this way, see, for the seed shape, what happens here? Phenotypic ratio is as follows: large, intermediate and small. These are the three types of phenotypic effects are produced for the seed size. So how many are large here is 1, which is having a homozygous dominant condition. How many are intermediate here? 2. And how many are homozygous recessive and producing small size seed? 1. Then what is the genotypic ratio? So here genotypic ratio is 1 is capital B, and 2 are capital B, small b, and 1 is small b. That is homozygous dominant 1, heterozygous condition 2, which produces intermediate phenotype, and 1 is a homozygous recessive condition. So here phenotypic ratio is 1:1, genotypic ratio is also 1:1 when the seed size is considered. But when the phenotype seed shape is considered, the phenotypic ratio will be 3:1, genotypic ratio will be 1:2:1. So when the same gene is controlling more than one character or the more than one phenotype, it becomes Pleiotrophy. So this is an example of Pleiotrophy. By this what we can conclude? ಇದರಿಂದ ಏನು ಗೊತ್ತಾಗುತ್ತೆ ನಮ್ಗೆ ಹಾಗಾದ್ರೆ? So dominance of an allele or the dominance is not a autonomous feature of any gene or the allele, but it depends on the phenotype or the product which we choose to examine. When the seed shape is considered, here one allele shows dominance over the other. When the seed size is considered, when the seed size is considered, there is no dominance. See, for example, in heterozygous condition, intermediate phenotype is produced. That shows codominance. Both are getting expressed. As a result, intermediate phenotype is produced. Or it also shows incomplete dominance. Dominant allele is incompletely suppressing the expression of the recessive one. So, the intermediate one is produced. So, here, dominance is not an autonomous feature of any allele of the gene, but it depends on the type of the product which we choose to examine. So, by this what we can understand? So dominance is not an autonomous means inbuilt feature of an gene, but depends on the type of product or the gene product which we choose to examine. See, this interpretation is very important in order to understand the dominance. Is it clear? And that is very important for the NEET examination and CET examination. Remember, so in this way, the gene coding for the seed shape and the seed size, that shows pleiotrophy. There is more than two effects, or the more than one phenotypic effect will be controlled by same gene. So it is an example of the pleiotrophy. Make a note of these points now.